Hi, I'm Destiny, and today we're going to cover prokaryote transcription. So transcription is the process in which a gene's DNA sequence is copied or transcribed to make an RNA molecule. RNA polymerase is the main transcription enzyme. Beginning with RNA polymerase, it is a core enzyme, meaning it has subunits including alpha-1, alpha-2, beta, beta-prime, and omega, or a holoenzyme if it also includes sigma factors to aid in this enzyme's functionality, such as heat shock genes, flagella and chemotaxis genes, or extra cytoplasmic functions. So now we're going to jump to the prokaryotic promoter region. So working from the left, which is more upstream, to the right, which moves you downstream, we come across our -35 and -10 regions. These are referred to as consensus sequences as they help recruit RNA polymerase. These regions hold complementary sequences which give RNA polymerase permission to bind to them so it can begin transcription at the +1 region. So now let's look at the entire transcription process. And it could have been broken down even more, but this is pretty much a summarized image of what's happening. So starting with initiation, which is the bonding process, we get RNA polymerase to bind to the template DNA strand near the start site of transcription by using the sigma factor right here to help locate the consensus regions on the promoter, which I kind of approximated and drew in. So once this occurs, the DNA strand unwinds and the template strand, specifically the 3 to 5 strand, is available for binding. So once RNA polymerase leaves the promoter, it begins transcription and the process of elongation. Finally, we finish up with termination of transcription, which is determined by what we call the rho factor. This goes one of two ways, either rho independent or rho dependent. So rho in this case is a protein. If independent, that means rho is not present, and likewise, if dependent, rho is needed for termination. In rho independent termination, RNA is physically modified to where it can no longer transcribe, and termination occurs. The RNA strand, once near termination, will form a loop-like structure, which is commonly referred to as a hairpin loop, because it kind of looks like a bobby pin. In rho-dependent termination, the helicase protein rho terminates transcription. So rho acts as a helicase and unwinds the DNA RNA hybrid strand to end transcription. And these are my references.